So, without further ado, it is my pleasure to introduce Dr. Bill Petri. Dr. Petri received his MD Ph.D. from the University of Virginia and is currently at UVA as the Wake Hampton Frost Professor of Medicine and Vice Chair for Research of the Department of Medicine. His research focuses on the immune mechanisms of defense against different pathogenic infections, and he has served on numerous scientific panels and leads the Bill and Melinda Gates Foundation's PROVIDE study, which stands for Performance of Rotavirus and Oral Polio Vaccines in Developing Countries. He's won numerous awards throughout his career and was named one of Virginia's outstanding scientists in 2017. So today he will be talking to us about poliovirus as an example of an epidemic disease that has been brought under control. So please join me in welcoming Dr. Petri.
Hey, Rachel, thanks very much. And a shout out to Judy Gilmartin, who's on, it looks like is online. Alicia Gilmartin just got her MD PhD at UVA. So like one of her graduates we're really proud of. Yeah, normally I would ask you guys to just interrupt. So I kind of, it's a little bit harder, I guess, like with Zoom to do that. And so I'll talk like for a little while, but I'm really hoping that we'll have like a discussion because it's much more fun to talk about things than for me to talk to you, so to speak. So we just met the WHO Polio Research Committee this morning, and so a lot of what's being discussed now is the disruption, because of new coronavirus, and as we were just saying like earlier, the polio eradication campaign, in addition to the expanded program of immunization, is halted for the time being, and whether that time being is a month or 6 months, is not clear to anyone. And a lot of the planning today at WHO was how to resume immunizations, and they really started thinking that it has to be negotiated country by country, of course, because WHO has like no power to dictate anything. But they have like, stockpiles of all the vaccines. And so they can go in and they won't just be immunizing for polio, but they'll be immunizing for measles and for tetanus. So it's really like, I was talking to my wife, Mary Ann, this afternoon saying, well, does this really make sense that we're going to put all these children in the world at risk for all the other infectious diseases in order to participate, have social distancing? And it really is like a difficult trade-off or discussion, which maybe we can chat about that after a few remarks. But I'm this person, so quarantine is like the best thing in the world for me and everything. I couldn't be happier. So I was like, they say like you can tell like whether you're introverted or extroverted based on what do you really like to do if you have just like some time to yourself. And so this has been just wonderful for me. But apologies for all the extroverts that are in quarantine. I'll tell you a little bit, but the history of polio, I think, is interesting. This is an Egyptian priest who had polio. The paralysis is asymmetric, and so it's not usually both legs, and that's one of the reasons we can be relatively assured that that's what his affliction was. This is the polio epidemic in New York City, and it's really interesting. You go back and read, and the practice of like social isolation and really started back with polio epidemics. My late mother I remember her telling me like growing up in East Greenwich, Rhode Island, how in the summertime with the polio epidemics, they would close the pools and the movie theater would be closed. And I remember her telling me this as a child, just thinking, wow, that's really incredible. We will never live in that age again. And so it's quite remarkable in a sad way that we're right back there. There are 3 different kinds of poliovirus, and to cut to the chase, of the 3 types, type 2 and type 3 have been completely eradicated from the world. So the polio eradication campaign is successful, but not without challenges. And so there still is type 1, and it's 2 Australian scientists, Gene McFarland and McFarland Burnett, Gene McNamara, excuse me, that were responsible for identifying that there was 3 different types. And then these 3 men won the Nobel Prize for the development of being able to grow viruses in cell culture. Since viruses aren't totally independent living beings, they need to like use like the host cellular machinery in order to replicate. It's different from bacteria or parasites that you can grow in a rich media and lab culture. And so Weller, Robbins, and Enders figured out that if you took like a human cell line that had the polio virus receptor on it, you could take the filtrate from the spinal cord of a primate that had been given polio, and then you could infect those cells and replicate the virus. And so that discovery made in 1948, and with really astonishing speed, by 1955, Salk had developed the first polio vaccine. So we were talking today about like having COVID-19 vaccines available probably, next year, probably like next winter, which is like just like lightning speed. And like talking, my wife's, sophisticated family physician, we say like, that's forever. Like how can it take like, 9 months to make a vaccine? But this was like light speed. Could go from 1948 to 1955 to develop the first polio vaccine. They actually did a study with 1.8 million children to show that the vaccine worked, because the paralysis was rare enough. It was like 50,000 cases a year in the US that you had to have a huge study in order to have enough children in the control group get polio to know that the vaccine worked. And when the trial results came out, there was a nationwide celebration, and Salk was actually invited to the White House by Eisenhower. And a lot of the support that we have today for the National Institutes of Health came from the public realization that if you invest in science, that this is one of the payoffs, that you didn't, if you were a parent, you didn't have to worry about your child, succumbing to an epidemic of paralytic polio. Then Albert Sabin developed the live oral vaccine. And both of these vaccines, both the Salk and the Sabin vaccines, are used today for global eradication. And the oral polio vaccine was not well accepted in the US. And so Sabin actually did most of the clinical trials in the USSR. And so but very, very important clinical trials. And we use both of these vaccines today for global eradication because they do different things. And so this inactivated polio virus vaccine that Salk made, we call it IPV for inactivated polio vaccine. That's a shot that you get in your arm. And that's what everybody on this phone call has received. If you're as old as me, you also got this oral polio vaccine. But this prevents paralysis because you get really good antibodies in your bloodstream against polio virus, and that prevents the virus from going from your intestine, where the infection starts, to your spinal cord. But it doesn't prevent paralytic polio. It doesn't eliminate, polio virus infection in the intestine. And since there's 100 children that are infected for every one child that gets paralysis, you really need to be able to have immunity against infection in your intestine. Otherwise, there's always going to be like some low smoldering level of polio. And for example, in the United States, we could never totally eliminate polio with the inactivated vaccine, but using that along with the oral vaccine, we were able to eliminate it. And then that's also how types 2 and 3 have been eliminated, is predominantly because this oral polio vaccine gives you a level of immunity that prevents fecal-oral transmission. So the way you get this infection is from fecally contaminated food or water or fomites, people's hands, and so you really need to be able to have immunity in the gut or in the intestine, and that's what Sabin's vaccine does. So, and the way that it does that, Pere Ogra, who was a professor at University of Buffalo, discovered that when you give the live oral vaccine, you get an immune response in the intestine. And this is like a really complicated slide, but this is with the Salk inactivated vaccine. This is looking at IgA antibodies in the duodenum or the small intestine. And you can see the child is immunized. You get a really nice level of antibodies in plasma or in serum, but you don't get any at all in the intestine. Whereas when you give this live oral vaccine, this is like the sugar cubes that the older members of this audience swallowed as a child, you get like that serum antibody response, but you notice here you're also getting antibodies in your duodenum. And it's these antibodies in the duodenum that protect you from infection. This is another nice thing about the history of polio. This is how we realized that there was two different immune systems. There's a systemic immune system, which the Salk vaccine activates. And then there's a mucosal immune system, which is in the gut, in the lungs, in the genital tract. And that's what the live virus vaccine activates. So I'm getting some chats. So let me see if I can. Good. So just someone just commenting that Dr. Robbins was at Case Western Reserve as Emeritus Dean. I did my P's residency at Rainbow Babies. I did like my medicine residency at Case Western, so we hold that in together. And then also mentioning like the sugar cubes. I think that the oral polio vaccine was eliminated from use in the U.S. I think around 1970 or so. So this is what's getting interrupted today, is the expanded program on immunization is stopped at a standstill right now. And this is like a wonderful program that not only gives like drops of oral polio vaccine, but gives tetanus vaccines to children, measles, and all this is being interrupted right now, unfortunately. Sorry, another comment here, excuse me. So, yes, so in Wahoo in Cleveland, all right. Sorry. So Rotary, if you're a member of Rotary, you're my friend for life. The Rotary Club has been absolutely amazing in their support of polio eradication to the tune of like almost $2 billion. Incredible contribution from a single volunteer group. So 12% of the total cost of the eradication campaign. So really, really something. And 1988 is when the UN made the decision, well, let's eliminate polio from the world. And at that time, there was 1,000 children every day that were paralyzed with polio. And so you can see that today that number is 1. So it's already had like a huge impact. We're not finished yet, which is part of the story. 1994, the Americas were certified polio-free. One of the questions this morning to the WHO director who runs polio vaccination was like, well, what's going on in the Americas with the interruption of vaccination due to COVID? And he says, oh, well, the Americas aren't part of WHO anymore. Sort of like tongue in cheek. So, but kind of sort of sad too. So, but 2002, Europe is certified polio free. 2011, the last case of wild polio in India. One of my fellows, Rajat Madan, got his MD in a medical school in India. And he remembers, like, on the national immunization days, the entire school shut down and everybody from the custodian to the dean were out giving oral polio vaccine in the community. So an incredible public health effort to eliminate polio from India. And then this is the last case of wild polio in Africa. And so 2016, and so very, very soon we'll be able to announce like that Africa is free of wild polio virus. So that's really wonderful. So the only place that there's wild polio right now is in Pakistan and Afghanistan. And this just shows you here's Karachi in Pakistan right here. So this is like the last challenge for wild polio virus eradication. And they're actually trying to figure out, like, you know, what unique solutions do they need to make this work. And a big part of it is negotiating with the Taliban, which has been ongoing, WHO. And I think there's been like a lot of progress as far as like getting the Taliban to promote polio eradication. Unfortunately, again, this is all going to be sort of on the back burner because of COVID-19. And so while there was like, you can see about 100 cases of wild polio in Pakistan, Afghanistan. Here's Afghanistan up here, Pakistan down here. That presumably is going to increase because all the immunization activities are at a standstill. So now the picture had been mostly like very rosy, but we've run into one problem, is that this live vaccine that Albert Sabin developed, he made the virus so that it was not neurovirulent, that it didn't cause paralysis, by just growing it in the laboratory in cells for, you know, passages and passages, and it lost the ability to be neurovirulent. And so the problem is about 1 out of 1 million doses, the virus reverts. It partly gains mutations that make it more neurovirulent, and then it can also genetically recombine with similar viruses that are called enteroviruses that are also present in the intestine. And this has become now a very major problem with polio eradication. And so what we're seeing now is not just wild polio 1 in Afghanistan, Pakistan, but we're now seeing circulating vaccine-derived poliovirus 2. It's this bad nomenclature, but it's called CVDPV2. And so each of these green dots is a child who was paralyzed due to the Sabin type 2 virus reverting from just being an innocuous component of the vaccine to actually causing paralysis. And so this is spread through Africa and even into Papua New Guinea and Indonesia, and I guess into Myanmar as well. And so this is like the new challenge now. And so Africa, there's no wild polio at all, but we have circulating strains of the vaccine virus that are causing paralysis. And so it's like the oral polio vaccine that Sabin made is like this two-edged sword. It's the one way to eradicate polio because it makes your intestine immune to polio, but at the same time, it has this rare ability to revert to be neurovirulent. And unfortunately, in areas where there's very poor levels of vaccination, as there are in like lots of sub-Saharan Africa, plus there's poor sanitation so that you have fecal oral spread. The combination of those two things engenders things like this happening, where you have a revertant virus being spread. So this would never happen in the United States. Number one, because our levels of vaccination are so high. And number two, because our sewage system is so good. And so you don't have raw sewage in contact with people. But you have both of those problems in sub-Saharan Africa, low levels of vaccination and poor sanitation. And so that has enabled these revertant viruses that are called this CVDPV2 to spread. And that's the challenge. And the only way today to combat that is by giving more of the same virus vaccine, more of the Sabin vaccine, because the inactivated Salk vaccine will help to prevent paralysis, and that's being given too, but it does nothing to eradicate this circulating vaccine-derived polio virus. So we're fighting fire with fire. I was sharing with Rachel, we have an editorial coming out about this in Science like later this month about this problem. And we went all throughout the page proofs. They made sure there's no typos. And then I got an email last night from this editor of Science saying, I couldn't get this right. This can't be right. They're not actually using the same vaccine that causes this problem to solve the problem, are they? And I said, oh no, you've got it exactly right. That's where we're stuck right now, is the only way to combat this is just to do more and more immunizations, try to raise the level of herd immunity, but with the knowledge that as you use this vaccine, you cause more cases of vaccine-derived polio. And so there is a solution that's coming out. And this is thanks to Bill Gates, that he has funded the making of a novel or new oral polio vaccine type 2, which is going to be much less likely to revert to neurovirulence. And so this is like for the, the molecular biologists in the crowd, but this is called the 5' untranslated region of the poliovirus genome. So the virus is this long strand of RNA, and it's actually the same sense as messenger RNA. And so when this RNA gets into a cell, the human ribosomes that make protein out of RNA bind here, and then they translate into make all the viral proteins. And so your host cell machinery is making this virus. And that's, of course, that's what Ender and Wellers got the Nobel Prize for, was like discovering, well, you could like use host cells to grow this virus because they would provide everything the virus needed, including a ribosome to bind right here. Well, what happens with that vaccine-derived polio that causes paralysis is you get mutations right here that make the virus much more potent in its ability to have the proteins translated. And then a second thing happens, which is that this part of the virus recombines with other enteroviruses that are in the human intestine, and then you get a neurovirulent virus. And so with the Gates Foundation support, they've mutated a site right here where recombination commonly happens, and they've also changed this untranslated region in a way that makes it much more difficult to be reverted to neurovirulence. And so this will actually, we heard this morning that this vaccine will be in use in 2021. So about the same time that maybe an emergency COVID-19 vaccine. And so the thought is that this will solve the problem. Now, Beth Kirkpatrick at the University of Vermont, runs just like a world-class vaccine center. You guys should all be like really proud of that in Vermont. And Beth is taking a different approach, is that she's taking the inactivated polio vaccine and she's adding what's called a mucosal adjuvant to it. And that's something that'll help this inactivated vaccine to give you that mucosal IgA antibody response to protect against not only paralysis, but infection. So if this doesn't work, there's every indication that this should be a good strategy, but you don't know until it's done. Beth Kirkpatrick is like right next with like the next approach to this, which is to take the inactivated vaccine and make it so that you get immunity in the intestine. And so that would be just like phenomenal. And you'd never have the risk again of what we're faced with in Africa right now. So just to close, and then I hope there'll be like lots of questions. So back to coronavirus, and I don't know if you guys have been following this, but there's a thought to use the oral polio vaccine to protect against COVID-19. And the idea is that vaccines stimulate what's called your innate immune system. So you have two different parts of your immune system. The acquired immune system is what makes antibodies, and that's like the IgA antibody in the duodenum. The innate immune system acts before your acquired immune system even has a chance to do anything. And like lots of organisms do perfectly fine without antibody, without T cells, like Drosophila, the fruit fly, does just fine without that. But the reason like Drosophila can survive without antibody is it has this thing called the innate immune system, which is things like neutrophils and macrophages, other kinds of white cells that can non-specifically attack and defeat invading organisms. And so this is like, the first couple days of an infection, you're not going to be protected by antibody because it takes like about a week to develop antibody. And so evolution has given you another arm of the immune system called the innate immune system. And there's a ways to non-specifically activate the innate immune system. So like one infection will activate the innate immune system in a way that will protect from other infections. And so there's actually going to be a clinical trial where thousands of individuals will receive the oral polio vaccine and see does that, by activating the innate arm of the immune system, can we protect against COVID-19? And we've actually done some of this work actually with Beth Kirkpatrick in Bangladesh, What we did was we actually randomized children that had already been vaccinated against polio to give them one boost. And the boost was either with the oral polio vaccine or with the inactivated polio vaccine, the solid line. And then we're doing this study for a different reason, to look for the best way to vaccinate against polio. But since we had these two arms, we looked to see, well, is this really true? Does this vaccine stimulate the innate immune system, protect you from diarrhea? And that's actually what we saw. So we looked, for example, this is Shigella diarrhea, the number of children that are getting Shigella diarrhea versus the number of days since they either received the Sabin oral vaccine or the Salk inactivated vaccine. I think you can see there's far fewer cases of Shigella diarrhea in children that got this oral vaccine, even though there's nothing in this oral vaccine that looks anything like Shigella. So this has nothing to do with antibodies. This is strictly from activating the innate immune system. So maybe there's something there, and we'll find out through a randomized clinical trial. So that's like my last slide, and I would love to hear your guys' thoughts and discussion. And thank you so much.
Thank you, Bill. It was a phenomenal presentation. Beth is actually my department head, so small world.
Yeah, Beth is like really like one of the smartest women I've met, next to you, Rachel. So.
Thank you. So I guess I'll ask the first question is, how do you see what's going on right now with the COVID-19? How does that really change the outlook of the polio vaccine being given? And really, how much does it delay eradicating polio?
Yeah, so Michelle Safran, who is the WHO person in charge of polio eradication, this morning was saying like that it's clearly there's going to be more children paralyzed in the next year because of the interruption of the polio campaign. Yeah, and then there's going to be more children that are going to die of measles too. I mean, this is like huge, big deal to interrupt childhood immunization. I don't see any other options right now in the situation that we're in, and so it's more like they're really like trying to figure out how do we like restart things, and it's going to be complicated. It's going to be like each country's going to be like slightly different about how you do that.
And I guess to follow up on that, do you ever see with such as the Ebola outbreak or any other outbreaks before, has it ever affected the vaccine as much as it is right now?
Yes, there's been major hurdles with vaccination, and it's usually like in countries where there's political instability. And so you look at like Nigeria, for example, was like the last country in Africa where polio was eradicated, and that was a lot due to Boko Haram. That's the terrorist group that kidnapped all the young women. They still control large parts of northern Nigeria. And so those were out of bounds for even like surveillance to try to identify where there are children with acute flaccid paralysis or polio, and certainly out of bounds for vaccination. And then again, like political solutions have been reached so that the vaccination is happening throughout Nigeria now. And of course, we're seeing this also in Pakistan, Afghanistan, both from ISIS and from the Taliban. And again, like negotiations now with the Taliban, before COVID-19 happened, were very encouraging to be able to promote protecting that country from polio. So it's almost like where there's political instability is where there's going to be problems with public health initiatives.
Excellent. Are there any questions from anyone else?
Yeah. Yeah, I guess Raharja, I guess. Yeah, please, yeah.
Yeah, actually, I don't know why my name shows up like that, but my name is Ryan Hargraves. So.
Oh, Brian. Okay, hi, Brian.
Yeah, I've got to fix that. Anyway, thank you so much for your talk. So, and maybe this is kind of a revisionist history question, but let's say hypothetically you were part of a leadership team that had the opportunity to influence the reaction to the pandemic in a specific locality or state or country, you could turn time back, say 4 weeks. What steps might you encourage based on best practices in the context of what you know about slowing or ceasing the spread of a pandemic?
Yeah, we know, and Brian, that's a great question. I think this is like what Tony Fauci is really kind of like outlining now, what will be our response after the pandemic settles down. And he's really making the point that we can't stop social isolation right now because we don't have the manpower or the diagnostic capability to identify most cases of infection. Because what you need to do, as all of us knows, you need to identify the cases and isolate them. You need to trace the contacts and have the contacts in quarantine for 14 days. And so in order to do that, you need to have like wonderful, much more robust testing abilities than we have today. You need to have like fewer cases too, because it's just like when there's 1 million people infected, that's probably 10 million contacts. And there's no way to do the case identification and contact tracing that you need in order to keep the epidemic down. The social isolation is really interesting though, because the reproductive number for COVID-19 is estimated to be 2.2, which just means so if I had COVID-19 on average, I'm going to infect 2.2 other people. Since social isolation, that number has dropped to 1.1. So now if I get infected with social isolation, I'm only going to infect, 1.1 less than one person, a tenth of a person. So if there's 10 people in social isolation, there's one additional person that's going to get infected. So yeah, but so if I had to do it all over again, what would have been done like in Wuhan would have been a very, very early case identification, contact tracing, and on top of it, social isolation. I think we could also see like the cat was out of the bag by the time it was realized what was going on there. Sorry, very long answer to your question, Brian.
No, thank you.
So Catherine has a question.
Yeah, Catherine.
Hi. So you mentioned the mucosal adjuvants in the vaccine, and I was wondering if the mucosal adjuvants effects were local to the duodenum, or if they stimulated the mucosal cells throughout the body. So would that indicate a vaccine that would be inhaled more like the flu mist used to be, or still a shot like we're used to?
Oh no, that's a great question. I hadn't thought about it, and I don't know the data for this particular adjuvant. One would expect that the mucosal immune system is more the same than different if you look at the gut and the respiratory tract and the genital urinary tract. And so I would think that yes, one mucosal adjuvant would give you activity at all those sites. We're actually working on a COVID vaccine. We have a different set of mucosal adjuvants that work very well, but we're doing it for a gut infection, a parasite called Entamoeba histolytica. But we're actually vaccinating mice now, and we're going to look in the lungs to see are we getting an IgA response. So Catherine, the same sort of thing that you're suggesting is like if you stimulate the mucosal immune system in one site, you should activate it in all sites. What Beth Kirkpatrick is doing at UVM is actually to inject this mucosal adjuvant, which has been done before, so it's known that it's completely safe. But even like giving it like as a shot in the arm, it has the ability to activate this mucosal immune system. And thanks for that question.
Great, thank you.
And Kirk has a question.
Yeah, Kirk. Yeah. Or you may be muted. I'm not hearing you, Kirk.
Kirk, do you want to try again?
I can read what he put in the chat. So,
Yeah.
Are there currently cases of polio in the U.S.? What does the rates look like?
Yeah. So Kirk, fortunately, there's no polio in the Americas at all. So North or South America, and And for a number of years, the only cases in the Americas were due to the use of the oral polio vaccine. And so it's the same issues that's happening in Africa right now, that we would have 2 or 3 cases of paralytic polio each year, and it was because of the vaccine virus reverting. And so that's the reason that the oral polio vaccine was removed from the US, was to prevent those rare cases of paralysis. And again, like we were in the situation with such good sanitation and such high levels of immunization, we no longer needed the oral polio vaccine. So a different situation than in sub-Saharan Africa today.
We have other questions.
I have a question. When is your last frost-free day? When are the gardeners out?
Someone will have to answer that, but I feel like it goes until May.
Oh, okay. Yeah. So just to rub it in, it was a week ago, so the garden's all in. So.
What's a garden?
Elizabeth has a question.
Yes. Hi, Bill. I do have a question. I confess to really having thought that polio was gone and then it had been eradicated. And I think that is because, as you were talking about in the Americas, it's just, I know my children received vaccinations, and it's just not something I've heard of, honestly. So I'm so glad to know that you're working on something, and that the Gates Foundation in and of itself is funding something that is so close to being removed from society, but that is still there and prevalent. The research that's being done right now, how long do you think, if the Gates Foundation and others continue, you're talking about the Rotary, which is phenomenal, once you get back and being able to do what you're doing, do you see an end that you really could say, we will be done and this will be removed? Is there a time frame?
There is a pre-COVID-19, the time frame was like 5 years to complete eradication. And again, like, so the only place, so the wild polio eradication is almost done. So there's 100 cases a year of wild polio in Pakistan, Afghanistan. Africa is wild polio free now. And so it was a really, really wonderful story until about 2 years ago, when it became apparent that through sub-Saharan Africa, we're having this problem with vaccine-derived polio. And I think that the positive part about this is that the new polio vaccine will be released for emergency use in 2021. And so if this interruption of vaccination is not long, we should be able to eliminate vaccine-derived polio in Africa. And then with the negotiations with the Taliban, and with a kind of a revamped program in Pakistan, Afghanistan, combat it there as well. The real threat is, like with COVID-19 going on, well, that the program will stop. We're so close, but if you stop, then wild polio will spread throughout, areas that are not well immunized, just as we're seeing like COVID-19 spreading. And it's an expensive program. It's about, I think, somewhere to 10 to 20% of the WHO budget is spent on polio eradication. Tremendous contributions from the U.S. government through the CDC and through the Rotary Club. So it's a very, very expensive proposition. The economists have calculated that long term, it's cheaper to follow this all the way through and eradicate polio than to have to care for children that are paralyzed. And so, but you sort of get campaign fatigue. So this has been going on since 1988, and so there's really like a lot of urging, like, to just sort of continue. Like whenever I meet like with our Rotary Club, I thank them and say, just thank you for like sticking with it as long as the Rotary has too.
So C. Townsend had a hand raised, and then Wendy.
Yeah. Can you hear me?
Yes, I can. Yes.
Oh, great. Hi, Charlie Townsend here. Seasonality. Can you tell us if there's a seasonality component to when people get polio? And then also give us your assessment of whether seasonality plays a factor with COVID-19.
Sure. There doesn't appear to be very much, Charlie, seasonality to polio, and maybe that's because it's mostly the disease now is in sub-Saharan Africa and Afghanistan and Pakistan. It was seasonal in the U.S., and so it was summertime epidemics. That's why my mom remembered her swimming pool in her community being closed. But there's less seasonality. It's interesting because flu, of course, is very seasonal in the U.S. We have flu season, but it's not seasonal like in Bangladesh. And all the reasons for that, I don't totally understand, because obviously, it gets cold in Pakistan, Afghanistan too, so you think there'd be some seasonality there. And it's, I think we're all waiting and we're hoping the coronavirus, this new coronavirus is seasonal. Certainly the common cold coronaviruses are, and that's thought to be partly just because in the wintertime we're closer to each other, where more people are indoors, more person-to-person transmission. Part the thought that the humidity and the temperature controls the viability of the virus in the air and on surfaces. So I think it's, I was going over the most recent modeling for COVID-19 with our lab group on Wednesday, and to me the biggest thing to remember was that no one knows, and the models are only as good as the existing data, and we just don't know what's going to happen with seasonality.
And Wendy has a question.
I think I'm unmuted. Can you hear me?
Yes, I can hear you, Wendy. Yes.
Okay.
No.
Yeah. Hi. So I wondered if you had any speculation about the use of the serologic testing that's emerging with COVID-19, any sort of utility in any dimension of the way the pandemic's playing out in the U.S. And I actually had another question that I had sent in when I registered, slightly different, but just I realized I didn't really know much about the polio epidemic in the U.S. and whether there were the kinds of disparities that we're beginning to see with COVID-19 prevalence in the U.S. now, whether that was true in the earlier polio epidemics. Thank you.
No, Wendy, I don't know if there was the same disparities with polio that we're seeing with COVID-19. It's a terrible problem with access to medical care in the U.S., as you're aware of. To me, the most dramatic thing is HIV, whereas we have all the medications, all the knowledge we need to virally suppress everyone. But yet, you look in Virginia, which is a very well-to-do state, only half of our patients with HIV are virally suppressed. And that's an indication of the problems with access to medical care, adherence to medical care. So it's a big issue, what's called implementation science. And so something that works, how do you get it out and get it used properly? And so we're for sure seeing that with COVID-19. The antibody tests, we're actually working on that in our lab as well, will be, I think, very valuable, because it looks like that a specific antibody response against the spike glycoprotein, which that cartoon with those red things that are sticking out of the virus, that's the spike of the virus. And that's what the virus uses to attach to the human receptor, which is called ACE2. And that part of the virus that sticks to the receptor has to be very conserved, because that's a specific, it's like a lock and key. And so antibodies against this receptor binding domain of the spike are neutralizing to the virus. And so they prevent the virus from attaching or entering a host cell. And so as these antibody assays get better, they'll be able to measure specifically for that neutralizing antibody response. And then that should be predictive of immunity. Well, what's not known is how long lasting that will be. The very closely related coronavirus that caused SARS 20 years ago, those antibodies lasted for a year and a half. And so that's very encouraging, because the common cold coronavirus antibodies only last for weeks. So long-winded answer again, but I think that the antibody tests are going to be very useful for actually being able to identify immune states for this.
So we had a few questions when people signed up to hear this talk. So one is, what did the responsible municipalities take in helping to combat the polio crisis when it was here in the U.S.?
I'm sorry, Rachel, what could have been done, or?
Yeah, I guess what did cities do in terms of taking care of or what they did to combat the polio crisis?
Oh, thanks. Yeah, so it's very reminiscent of what we're doing today. And so social distancing, to the extent like that they closed down like major events, not to the extent that we're doing today where we're all trying to stay in our homes for the next several weeks. But again, like closing down the movie theaters, the swimming pools, the major sporting events. And again, it was seasonal, like Wendy's earlier question. And so that would just happen during the summertime when epidemics were happening. And it was also spotty, and so some communities would have an epidemic and other ones wouldn't. And I think we're seeing a little bit of that with COVID-19 as well, like that some places are much worse off than others. So.
And then someone had a kind of a fun question. Are you related to the person who invented the Petri?
I'm not. And so that person's name is spelled in name, but my family's name was misspelled when we immigrated. And so we're both families are from Alsace-Lorraine, but unfortunately no relations. An interesting story, Rachel, is that the Petri who invented the Petri dish didn't invent the bottom, like where you grow the bacteria. He's the person, if you put a top on it, that the little fungal spores won't settle out into and contaminate your bacterial cultures. And I think we're all very happy that Fleming didn't have a top on his Petri dish, because otherwise the penicillium mold wouldn't have landed and killed the streptococci that were growing on the dish, thus leading to the invention of penicillin. So.
And Catherine has another question.
So just to follow up from your discussion about polio and the response in the US where things were closed, was there a political debate about that, or is that more of a recent thing where the closing things is a very political thing in the current climate?
Yeah, that's it. No, and I don't know, Catherine. It really would be very interesting to go back and then try to understand more like what was being done in the 1950s. Yeah, I'm sure that there's still people that have like that oral history from 1950. So I think it must have been like just so frightening. I think we're all frightened right now, right, with COVID-19. And as a parent in 1950, to think that your child was at risk of of having paralysis, just out of the blue with like a summertime epidemic. And again, I think that just had a huge role in the support of biomedical research, when that fear could be taken away by scientists being able to grow the virus and then make a vaccine out of it.
Okay, and I think this is the last question, unless anyone else have other questions. So it's about after you have the primary immune response, there's the production of the IgM antibodies, which also results in the IgG antibodies. So is it automatic that individuals who are no longer infectious, and they're saying for the COVID-SARS-2, once they have these antibodies present? So essentially, once you have IgM and IgG, does that mean you're no longer shedding the virus?
No, it doesn't, unfortunately, because we know that those antibody levels, those antibodies develop within a few days of infection. But the average person that has COVID-19 is excreting virus for about 20 days or so. So, and as you might ask, like, well, these antibodies are protective. How can that be? Well, the antibodies can't get into the cells where the virus is replicating. And that's going to take what's called a cell-mediated immune response. And so antibodies alone are not going to be effective at clearing an established infection. It's going to need the combination of that and cytotoxic T lymphocytes, for example. But for a vaccine, what you'd want to do ideally is prevent the infection. And so if you can generate those antibodies in advance of exposure, then the antibodies will bind to that red spike glycoprotein in the cartoon, prevent the virus from even attaching to the human cells to cause infection.
And then I have one more question. So one of the things you were saying is with polio vaccines that you're actually getting infections from the vaccine. And I can just imagine in the US, if that was what was happening almost no one would be taking their kid to get vaccinated. So are you seeing the same in these other countries? Is it affecting the vaccination rate?
Oh, that's interesting. And I don't know the answer to that, Rachel. I know that there's been problems in the past where, the thought like the vaccine is being used to like sterilize like certain ethnic or religious groups and other sort of campaigns of misinformation. But I don't know the extent to whether there's any resistance to vaccinations. I think really just the opposite is that these mass vaccination campaigns are really well accepted in the community. I think like one good thing that will come out of COVID-19 is that the polio workers now are totally transformed into COVID-19 workers. And I think we were talking this morning at this meeting of the WHO was like that. So that's going to also help, I think, with future acceptance of polio vaccination, to see that these very same people that are interested in vaccinating your child against polio are also helping you with COVID-19. So.
Thank you.
So can I would just like to say like one last thing?
Yeah.
My oldest daughter, Sarah, went to UVM for 4 years. She's now a veterinarian in Portland, Oregon, and she had an absolutely wonderful experience at UVM. And so thank you, everybody that was involved in giving her such a great experience.
Well, I'm glad she loved it here. Besides the snow, can say I also love it here. But if there are no more questions, I'd just like to thank you again, Bill, for taking the time to give us this talk and answer all of our questions.
Well, Rachel, and thank you so much. It's great to see you again. So
good to see you as well. So with that, we'll go ahead and end it. Hope everyone's staying safe, and hopefully I'll see you in the near future.
Great. Thanks.
Thank
Bye-bye.
you, Bill. Thank you, Rachel. This was awesome.